Hello everyone and welcome to the new session of the biology lecture series, and today we'll be discussing the NCERT Example problems for the topic evolution. So in the last session we have discussed the multiple choice question, and in this session we'll be discussing the very short answer type questions. So I would like to mention that all the questions in this session are completely NCERT based. So let's start with the first question. So first question is, what were the characteristics of the life forms that had been fossilized? Okay, so here you have to tell what were the features, what were the unique features of the life forms that has been fossilized. So you have to tell the features of those life forms or the living organisms whose fossils have been discovered. All right. Now, let's talk about this. What are fossils first? So fossils, they are formed and they are preserved over time, and all the organisms, they are not likely to be fossilized. So all organisms cannot be fossilized. There are certain unique features, okay, and if the organism have those features, only then they can be fossilized. So it is based, means the fossil formation is based towards organisms with hard parts such as bones of the vertebrates. So you know what are vertebrates? Vertebrates are those organisms that have the vertebral column, they have the skeletal system. All right. So one criteria of the organisms is the presence of bones, such as in vertebrates, or the calcareous exoskeleton of invertebrates. So invertebrates are those organisms which lack vertebral column. Okay, so the organisms that have bones or they have calcareous exoskeleton, those organisms can be fossilized easily. Now let's talk about the explanation. So this is the solution. The organisms with hard parts, okay, hard part means those organisms which contains the bones and the calcareous exoskeleton. So these organisms are likely to be fossilized than those who do not have such parts. So the harder the material, so you know bones and the exoskeleton, they are very hard. Okay, so the harder the material, better it would be preserved, okay, as compared to the soft part. All right, so the hardened material, it is preserved, whereas the soft material or the soft part, okay, they doesn't appear as fossil, so they cannot be fossilized. For example, birds and pterosaurs, okay, they have very light bones, okay, and they are hollowed out, okay, and they are specialized for the flight. So these birds and pterosaurs, they are not easily to fossilize because they have very light bones, they have soft parts, okay, the bones are hollow. Hollowed out means their bones are hollow, and you can see why they are hollow, because they have the ability to fly. Okay, if the bones are very hard, they are very thick, they won't be able to fly. That's why the bones are hollow in birds and organisms like pterosaurs, which is extinct now. So these are the criteria for the organism to be fossilized. One is the presence of, you can say, bones or the hard structures, or the presence of calcareous exoskeleton in invertebrates. And the organism that have soft parts, okay, such as hollow bones, okay, or very light bones, these organisms cannot be fossilized easily. Now, so the organism with this soft bone, they have sparser fossil record, means less fossil records as compared to mammals whose bones are partially mineralized during the life. Okay, so bones, you know, it a very hard structure, so it is partially mineralized and the organism can be fossilized. Now let's talk about next question. Did aquatic life forms get fossilized? All right. Now, if yes, where do we come across such fossils? So here the question they are asking, terrestrial life forms, okay, they can be fossilized. Whether the aquatic life forms like fishes and all, they can be fossilized or not. So if yes, so these fossils can be found where. Okay. Now, this is the thinking process again. I've given the geological changes that took place over the time. They transformed many water bodies into solid rocks and mountains. All right. So what does this mean? This means there was a lot of geological changes, and because of that, the aquatic bodies or the water bodies, they were transformed into solid rocks and mountains. Okay. So fossils of the aquatic organisms are more likely to be found in the mountains. You must be confused why the fossils of the aquatic organisms are found in mountains and not in the seabed, you can see, or on the surface of the sea. Okay. Now let's discuss the answer here. So, yes, aquatic forms of the life, they do get fossilized. In fact, there are more aquatic than terrestrial fossils. Okay. So the aquatic fossils are more as compared to the terrestrial fossil. Now, such fossils of sea creatures are found in mountains as opposed to deep sea beds. So this I've already told you. The fossils of the aquatic life forms, or you can say the sea creatures, they are found in mountains as opposed to the deep sea beds. Now let's see how the fossils are present in the mountains. So this is because the rocks in which the fossils are found, okay, they used to be at the bottom of the oceans. Now what happened? Due to the changes in the crustal plates, okay, over time, the ocean sediments were pushed up to form the mountains. Okay, this is clear. There were changes in the crustal plates, and because of this, the ocean sediments, they were pushed up to form the mountains. So that's why the aquatic or the fossils of the aquatic life forms are present in the mountains rather than the ocean beds. Okay, so this is clear. Now let's talk about the next question. So here question is, what are we referring to when we say simple organism to complex organism? All right. So here you can see in the header, I have given you the concept on which this question is based. So in each slide, I have given you the header and the footer. So footer, you can see the concept on which this question is based. Now let's discuss the answer. Now these terms are used to classify organisms according to their evolutionary history. Evolutionary history, what does it mean? Means phylogenetic inheritance pattern. Okay, so phylogeny means evolution only. Okay, or you can also say these terms are used to classify organisms according to their phylogeny or phylogenetic relationship or evolutionary relationship. Okay, so now if you talk about the simple organism refer to those organisms that have simple structural and functional organization and are considered primitive. So you already know that the simple organisms, they have very simple organization, they have very simple structure, and they have very simple functional organization, and because of that they are considered primitive. Whereas the complex organisms refer to those organisms that have higher and complex level of structural and functional organization. Okay, now these are more advanced and said to have arisen from simple organism. Okay, this is clear. Now let's talk about next question. How do we compute the age of a living tree? Okay, so here a living tree is given to you, suppose. Okay, now you have to compute the age. You have to calculate the age of that living tree. So there are a lot of methods to calculate the age. I will tell you one of the method. Okay, how to calculate the age of the living tree. So This one is a very simple method. To estimate the age of the living tree, following steps are required. First one, you can measure the circumference first. Circumference of what? Circumference of the tree trunk at about 4.5 feet above the ground. Okay, so first you have to measure the circumference. Circumference means perimeter also. So these are both the same thing, circumference or perimeter. Okay, next question is, next point is, after calculating circumference, you can calculate the diameter of the trunk. Okay, so there is a formula. Circumference, or you can say perimeter, is equal to pi into diameter. So D is diameter, C is circumference, this is pi whose value is 3.14, this is the diameter. So if you calculate the circumference, you can calculate the value of D, which is the diameter. So here C divided by pi is equal to the diameter. Okay, so here how you can calculate the diameter? This is done by dividing the circumference by 3.4, which is the value of the pi. Okay, you can divide C by 3.14. Okay, so then you will get the diameter. And then if you divide this diameter by 2, you can get the, or you can calculate the radius. So this is clear. Now you know the diameter. All right. Now, next point is, or next step is, you have to determine the growth factor. Now, what is growth factor? A tree's growth factor is the measurement of the width it gains annually. So this data is already available. Okay, you can check that tree's average growth factor. Okay, that must have already been recorded. You can use that. Now, the tree's growth factor can be seen from the data available or by measuring the rings of a dead tree from the same species. Okay, this is clear. This is how you can calculate the age of the living tree. Okay, now another method is, sometimes they use a borer. Okay, and then they insert that borer in the wood, and then they take out that a part of that wood, and then they calculate the annual rings. So by calculating the annual rings also, you can calculate the age of the plant. Okay, but that can be done for dead trees. Here they are asking how to calculate the, or how to estimate the age of the living tree. Okay, this is clear. Now, next step is, you can multiply the diameter and the tree species average growth factor. Okay, what you have to do in the end, you have to multiply the diameter with the average growth factor. Average growth factor, I have already told you, can be obtained from the previous database, and the so done calculation suggests the approximate age of the tree in years. So this is how you can calculate the age of the tree, age of the living tree. Okay, so simple, you can write diameter multiplied by the average growth factor. This will give you the age of the living tree. Clear? Now next question. Next question is, give an example or for convergent evolution and identify the features toward which they are converging. Okay, so you already know about convergent evolution. They give rise to what? They give rise to analogous organs. So what happens in convergent evolution? There are two diverse species and they converge together to form a similar species. Okay, suppose this is species 1, species 2. Okay, this is species 1, this is species 2, and then they converge together to form a similar species. Okay, this is clear. And this question is based on the concept of adaptive radiation. Okay, now let's have a look at the answer here. Now, when unrelated animals, or you can say unrelated organism, I've shown you some unrelated species, okay, they are converging to the same form or the structure. That's why converging evolution gives rise to analogous organs. And what are analogous organs? Which have same function but different structure. Okay. So when unrelated animals or the organism converging to the same form or the structure, that is very adaptive in their common environment. Okay, so this is known as convergent evolution. All right, or in simple words, you can say when unrelated organisms, they converge into a species or an organism that are very similar to each other. So they are converging. Initially, they were very distinct. Okay, they were unrelated. Now, what is happening? They are adapting to similar environment. Okay, and this give rise to a species that is very related. Okay, so this is known as convergent evolution. Example of convergent evolution you can see in Australian marsupials and placental mammals. So these are the example of convergent evolution. Now here both subclasses of the mammals, they have adapted to a particular food source, locomotion and environment in comparable ways. Okay, so both of these mammals, okay, Australian marsupials and the placental mammals, okay, they have adapted to what? To a particular food source, locomotion and the environment. All right, so they had the same food source or they feed on the same food, their locomotory habit is same, okay, and the environment is also quite similar. So they have adapted to these conditions. Now, their resemblance in overall shape, locomotor mode and feeding and foraging are superimposed upon different modes of reproduction. So they have different mode of reproduction, but they are similar in overall shape, locomotion, feeding and foraging, but they are different in reproduction. Okay. The only feature that accurately reflects their distinct evolutionary relationship. Okay. So they have different mode of reproduction and this shows that they are distinct evolutionary, or you can say they have distinct evolutionary relationship because they have different mode of reproduction. Rest all is same, like the overall shape is same, locomotion habit is same, feeding and foraging habit is same. Okay, so this is clear. So this is due to convergent evolution. Now let's talk about next question. How do we compute the age of a fossil? Again, we are talking about the fossil here. Okay, what is study of fossil? Paleontology. And the word paleo is, or you can say, represents the fossil. Now, the age of the fossil can be computed by radioactive dating, also called as radiometric dating. It is a technique based on a comparison between the observed abundance of a naturally occurring radioactive isotope and its decay products. Okay, so here what we use the decay rates. All right, so what is radioactive dating? So it is a technique which is based on comparison between the observed abundance of the naturally occurring radioactive isotope. Okay, so there is a naturally occurring radioactive isotope and it is decaying. Okay, so we take into account the abundance of the naturally occurring radioactive isotope and its decay products. Okay, so if you know the decay rate, you can calculate everything. All right, so based on the decay rate, you can do the radioactive dating. Now, among the best known techniques are radiocarbon dating, potassium argon dating and uranium lead dating. Okay, this is clear. Now let's move on to the next question. So uranium, if we talk about the uranium, in the previous question we were talking about the uranium. So here uranium decays into lead. Okay, so decay product is lead and you can do the radioactive labeling using this method. Now, what is the most important precondition for adaptive radiation? Okay, adaptive radiation we already know. Okay, it is a kind of divergent evolution. It gives rise to homologous organs. So, the phenomena in which new species arise by adapting to diverse environmental niches. And here we have a shared ancestor. You can say a common ancestor. This is known as adaptive radiation. Clear? So, the phenomena in which new species arise. First one is this. New species is arising by adapting to diverse environmental niches. So, there are different niches present here. Okay. So, there was a common ancestor. So, suppose this is the common ancestor or the shared ancestor. And then these, okay, this is showing divergent evolution. So, each one is adapting to its environmental niche. So, suppose there was a geographical barrier. Okay, so there was a barrier here. So, here there was some other conditions. Here it was some other environmental conditions. So, initially there was one species and it diverged into two species. This is species 1. This is species 2. Okay, but they had this shared ancestor. Alright, and now they are adapting to different environmental niches, or you can say niche, or different environmental conditions. This can happen due to geographical barrier. This is known as adaptive radiation. You can also call adaptive radiation sometimes as divergent evolution. Now, there are two primary mechanisms for adaptive radiation. Okay, first one is intrinsic changes. What does intrinsic changes means? Intrinsic changes that expands the availability of ecological niches for a lineage undergoing diversification. Okay, so intrinsic changes. What does it do? Intrinsic changes. It expands the availability, okay, of ecological niches for a lineage. So there is one lineage that is undergoing diversification. Okay, how it will go diversification? If there are different ecological niche available. Okay, so intrinsic changes, they expand the availability, or you can say they provide different ecological niche to a lineage. Okay, that is undergoing diversification. Now, second are the intrinsic effect. What are intrinsic effect? Intrinsic effect, they include the emergence of the novel environment. Okay, and colonization of the isolated land masses. So these are the intrinsic effect. This is very important. The emergence of a novel environment. Okay, so if novel environment or new environment or unique environment has been created, then the species will adapt to that. Okay, and a new species will form. Okay, so that species which can adapt to this novel environment, that will form a separate lineage, you can say, or that will undergo speciation and it will form a new species. Okay, so these points are very important for adaptive radiation to occur. Okay, first is availability of the ecological niches, and second is emergence of novel environment. The next question is, how do we compute the age of a rock here? Okay, so let's talk about the solution. Now, the age of a rock in years is called its absolute age. Now, it is determined by the natural radioactive decay of certain elements. For example, uranium. So, when uranium decays, it turns into lead. Okay, this already I have told you. Now, the parent atoms of uranium are converted into daughter atoms of lead over a fixed interval of time. Now, this interval is the decay constant. Okay. Now, the ratio of parent-daughter atoms changes in a quantity that can be measured. Okay. So, how the age of the rock can be calculated? First, by natural radioactive decay of elements such as uranium. So, uranium, when decays, it turns into lead. Now the parent atoms of the uranium, they are converted into the daughter atoms of what? Lead over a fixed interval. Okay, so this interval is fixed and you can use this to calculate the age of the rock. Now the ratio of the parent and the daughter atom. So parent atom will be uranium. Okay, and what will be the daughter atom? It will be the lead. So you can say the ratio of uranium by lead atoms, okay, changes in quantity that can be measured. And this is how you can calculate the age of the rock. Now, the radioactive half-life. What is radioactive half-life? The amount of time required for 1/2 of the parent atoms to be converted to daughter atoms. So, the radioactive half-life is used to calculate the age of the rock. So, simply, if you want to answer this question, you can say the radioactive half-life is used to calculate the age of the rock. And how do you define radioactive half-life? The amount of time required for 1/2 of the parent atom to be converted into daughter atoms. Okay, suppose 1/2 of the uranium, or you can say the amount of time required for 1/2 of the uranium to be converted into lead atoms. This is known as radioactive half-life of uranium. Now let's talk about the next question. In certain populations, the frequency of 3 genotype is as follows. Okay, so genotypes are given. This is homozygous allele, you can say, or the homozygous individual. This is the heterozygous one, and this is the homozygous recessive. This is the homozygous dominant. B is homozygous dominant. You can say capital B is homozygous dominant. Capital B, small b is heterozygous. Okay, and small b is homozygous recessive. And the frequencies are given here. 22% of homozygous dominant B allele, 62% of heterozygous B and small b allele, and 16% of the homozygous recessive. Now, we have to calculate what is the likely frequency of capital B and small b, means the dominant B allele and the recessive small b allele. You have to calculate their frequency. So this question is based on the Hardy-Weinberg principle. So this question I've already told you. So according to the Hardy-Weinberg principle, P square plus 2PQ plus Q square is always equal to 1. So this is the frequency of the homozygous dominant. This is the frequency of heterozygous, and this is the frequency of homozygous recessive. So this is homozygous dominant. Okay, and this is heterozygous. Again, Q square will be, this is homozygous recessive. Clear? Now, let's talk about the solution. Now here you have to use this formula. Frequency of a gene calculated by frequency of the homozygous gene, okay, plus the half frequency of the heterozygous. Okay, now we have homozygous dominant gene here and homozygous recessive. So the likely frequency of dominant B allele is, you can see, homozygous dominant gene plus the frequency of the heterozygous, or you can say half of the frequency of the heterozygote. So here it was given 22%. So here 62 divided by 2 is 31. And if you calculate, you get 53%. So likely frequency of B allele is 53%. Similarly, the likely frequency of small b alleles, okay, or homozygous recessive, you can see this is the homozygous recessive. The frequency of the recessive small b allele will be the frequency of the homozygous recessive Okay, and half of the frequency of the heterozygous. So this will be 16% plus 31%, which give you the answer 47%. This is clear? So to solve this question, you have to remember this formula. The frequency of a gene can be calculated by frequency of the homozygous gene plus the half of the frequency of heterozygous. And this you can calculate this formula using Hardy-Weinberg equation, which is p square plus 2p square plus q square equal to 1. Alright, this is clear. I've already told you what was p square, q square and 2pq. Now coming to next question. Now, among the 5 factors that are known to affect the Hardy-Weinberg equilibrium, 3 factors are gene flow, genetic drift and genetic recombination. Now what are the other 2 factors? So Hardy-Weinberg principle we have discussed in the last session. In the previous session we have discussed what was Hardy-Weinberg principle. So it states that the allele or the gene frequency remain constant from one generation to other generation in the absence of the evolutionary forces. Okay. So in the question they have given you these factors. So these factors are the evolutionary forces. So 3 they have given: gene flow, genetic drift and genetic recombination, and other 2 you have to mention. You have to answer what are the other 2 factors. So if there is no gene flow, there is no genetic drift, there is no recombination, if the population is large, okay, there is no inbreeding, alright, there is no overlapping genes, there is no mutation, there is no natural selection, then the population will follow the Hardy-Weinberg equilibrium, which means that the allele or the gene frequency will remain stable. It will remain constant from one generation to another generation. Okay. Now let's talk about the solution. The other 2 factors that affect Hardy-Weinberg equilibrium are mutation and natural selection. Okay. Now mutation, if we talk about the mutation, they are the sudden heritable change in an organism which is generally due to change in the base sequence of the nucleic acid in the organism's genome. Okay. So if there is mutation, that can cause change in the allele frequency, all right, and it can cause speciation, means formation of new species, because mutations, they are sudden, okay, but they are heritable changes in an organism. Okay. Now microbial experiments, this also we have discussed in the previous session. Microbial experiment show that pre-existing advantageous mutation. Okay, so there are already pre-existing variations, you can say, they in a population always. All right. So if we talk about the bacterial, say microbial experiments, so they show pre-existing variations which are due to the presence of these mutations, and these mutations are beneficial. Suppose in antibiotic resistant bacteria, you can see if you add antibiotic in the media, so already there are some pre-existing variations present in the population that are due to the mutations. So suppose due to the mutation the bacteria become antibiotic resistant, so that bacteria will be selected in the next generation and it will increase in number, okay, and it will become fixed, or that gene will become fixed. Okay, the antibiotic resistant gene will become fixed. All right. So the microbial experiments show that pre-existing advantageous mutations, when selected, will result in the formation of new phenotypes. Okay. Now, over few generations, this would result in speciation. All right, because here a particular character is being selected. Only those bacteria is being selected that have the antibiotic resistance. Okay. Now, thus resulting in change frequency of genes and alleles. So this is how mutation can cause speciation, and this is how it can lead to change in the frequency of the genes and alleles. All right, now if you talk about natural selection, so natural selection you already know, it is a phenomena by which organisms possessing heritable variations enabling their better survival they reproduce and leave greater number of progeny than their counterpart. So in simply, you can say that what happens in natural selection, which is a very gradual and a slow process. Okay, so first you have to remember it is a very gradual and a slow process. And here what is happening? The organisms or the species which is better adapted to the environment, that will survive, and it will leave greater number of the progeny than the counterpart. Okay, so the organism which can adapt to the change in the environment, that will be selected, and the other organisms or the other species that will get extinct or that will die. All right, so this is what is natural selection. Now, natural selection, there are so many types. We have already discussed stabilizing, okay, then directional, then disruptive. So these are the kinds of natural selection. So if you talk about stabilizing selection, what happens? Now, natural selection, it can lead to stabilization. In stabilization, what happens? In which more individuals acquire the mean character value. So this is what happens in the stabilizing selection. If this is the initial population, intermediate phenotype is selected. So this is stabilizing selection. All right. Then directional. In directional, what happens? More individuals acquire value other than the mean character value. So if you talk about directional, what happens? One of the phenotype is selected. Okay, so directional here. So this is being selected. This happens in directional. More individuals acquire a value other than the mean character value. Okay, so here the selection is happening in one direction. And what happens in disruptive or disruption? More individuals acquire peripheral characteristic. So here intermediate phenotype is not selected, but the extreme. So if this was the initial population, here you can see here at the periphery, at the extremes, these individuals will be selected. So this is disruptive selection. This is directional. This is directional and this is stabilizing. Okay. Now coming to the next question, what is founder effect? Now let's talk about the answer here. Sometimes a small number of individuals, they become isolated from a larger population to form a new population at some distance away from their place of origin. Okay. So what happens if this was the original population? There was some individuals here. Okay. So what happens? They become isolated. These individuals, they become isolated. Okay, from this larger population. Okay, so they will get isolated from this larger population. This is the larger population and they will form a new population. So here they get isolated. Okay, they get isolated. They come here and here they form a new population. Okay, here they are forming a new population at some distance away from their place of origin. So this is known as founder species. Now the gene pool of the new population differs from the source population here. Okay, it is possible that the change in the allele frequency is so drastically different in the new sample, means the new population, that they become a different species. So what happens? Now some of the organisms, okay, they isolate themselves, then they form new population, and there is change in the allele frequency here, and the change is so drastic that they form different species altogether. Now the original drifted population becomes the founders, and this effect is called as the founder effect. Okay, this is known as founder effect. And the original drifted population, they are known as the founders, and that's why this phenomena is known as founder effect. Now let's talk about the next question. Who among the Dryopithecus and Ramapithecus were more man-like? Okay, this is a very straightforward question. So, Ramapithecus were more man-like. They walked erect on their hind limbs, they ate hard nuts and seed like modern man, and had jaws and teeth similar to humans. That's why they are more man-like. Now, they arose from Dryopithecus. Okay, so Ramapithecus, they originated from Dryopithecus, which was considered to be a common ancestor of man and apes. So, Dryopithecus were more ape-like with same length of arms and legs. So, Ramapithecus, they are more man-like and they originated from Dryopithecus. Okay, so you can write. First were Dryopithecus, that were the ancestors of both, that were the common ancestors of both man and apes, and they evolved into Ramapithecus. Alright, that were more man-like. And then later on, most evolved species or the most evolved organisms are the Homo sapiens. Now, next question is, by what Latin name first hominid was known? This is also a very straightforward question. The first hominid were known as the Homo habilis. So after Ramapithecus, Homo habilis, okay, evolved. Okay, the first hominid was called as Homo habilis, and the brain capacity was between 650 to 800 cc, and they probably did not eat meat. Okay, this is clear. Now let's talk about the next question. Among Ramapithecus, Australopithecus, Homo habilis, who probably did not eat meat? Now, Homo habilis, let's talk about the solution. Homo habilis probably did not eat meat. This creature was the first human-like being with brain capacity between 650 to 800 cc. Okay, this is clear.